Good morning, my name is Kim and welcome to Science Today. We have two special guests for you this morning, Francis Crick and James Watson, the discoverers of DNA, or deoxyribonucleic acid, the building block of life. I'm interested to hear of your discovery. Can you provide our viewers with some history on your research?
There's a lot to say, but let's make this brief. James and I are the primary researchers of the DNA structure. We made our discovery in England in 1953.
Yes, Francis and I made a model of DNA, and it characterized DNA's double helix structure. The double helix looks like a long twisted ladder.
That is very interesting, and I have some deeper questions for you. Can you explain how DNA was made?
Well, the answer is quite complex, but I will attempt to simplify. Each strand of the DNA molecule was a template for the other. During cell division, the two strands separate, and on each strand, a new other half is built, just like the one before. This way, DNA can reproduce itself without changing its structure, except for mutations.
DNA is made up of 4 bases, phosphate, and sugar. The bases are adenine, thymine, guanine, and cytosine. It is also important to mention that adenine always pairs with thymine and guanine always pairs with cytosine. If these bases don't pair up correctly, a genetic mutation will occur.
I'm glad you said something about genetic mutations. That was my next question. So can you tell me a little bit more about genetic mutations?
Genetic mutations occur when something is not correct in the DNA replication process. There are 3 types of genetic mutations. There is insertion, deletion, and substitution. Insertion happens when an extra base is added. Deletion happens when a base is left out, and substitution happens when a base isn't paired correctly with another base.
When a genetic mutation occurs, there are 3 possible outcomes. These outcomes can be beneficial, neutral, or harmful. Genetic mutations.
Well, gentlemen, I'm sorry, but it's time for a commercial break. After the break, we'll talk more science.
Have you or a loved one been genetically engineered against your will? If so, you can get $1 million right now and the satisfaction of suing the company that put you in this terrible position. Call Lumby Law now at 1-800-GET-HELP.
Do you know that you can genetically engineer DNA? Well, you can. It's time to create your perfect dream child. If this sounds like what you would like to do, call 1-800-DREAM-KID or stop by our geneticist office to make your appointment today at 100 Rodeo Drive, Los Angeles, California, 12345. And remember, you know what's important, being perfect.
Wow, what a coincidence. My next question for you was about genetic engineering. In your expert opinion, how would you describe genetic engineering?
Genetic engineering is changing and manipulating genes. You can genetically engineer people, plants, animals, bugs, and more. However, genetic engineering is one of the largest ethical debates within the genetics community.
I believe that genetic engineering is the best thing that was ever discovered.
I disagree. In my opinion, genetic engineering is not only cruel, but interferes with Mother Nature. I'm shocked you support that, James.
Okay, let's leave this as you agree to disagree. As you can see, there is a lot of controversy about genetic engineering. Let's take a look at some photographs of genetically engineered things. Those were some interesting photos. Gentlemen, do you have anything additional that you would like to add?
Amazing!
Okay, before we conclude this interview, our viewers would like to know more about the research conducted by Ms. Rosalind Franklin at King's College in London. It has been said that she is also included in this discovery.
Rosalind's research was very important and led to our double helix discovery.
Yes, in fact, Rosalind passed away prior to our winning the Nobel Prize in 1962 for the discovery of the DNA double helix structure. Had she been alive, she too would have been presented with the award. Unfortunately, the Nobel Prize is only awarded to living people.
Well, I would like to take this opportunity to thank you both for spending time with me today and explaining the story of the DNA structure discovery. Your work was and is truly amazing.
Thank you, Kim, for having us.
Thank you. And thank you for being with me today. I learned a lot, and I hope you also learned a lot. Please tune in tomorrow for the scoop on how to stop bad dog breath.